So somebody asked me if I would introduce Judith, and it is a great privilege and honor to do so. And I realized in thinking about this that Judith and I have known each other probably longer than half of the people in the audience have been alive. But that's something we maybe don't want to make too much of a big deal. So Judith was my first PhD student, and she is single-handedly responsible for the fact that I got tenure, because it was her thesis work that really made all the difference in what we were doing and why and how. And I should say that Judith ended up joining my lab through a variety of strange circumstances. At the time, she was interested in developmental neurobiology, which she's continued in that vein. And she was working down at BU with a quite wonderful guy called Fred Lang, who had a lab looking at development in lobsters. And he was very, very, very sadly killed in a car crash, leaving 8 graduate students. And the then powers that be managed to find homes for many of them. And somehow or other, Judith ended up with me. And to this day, I do not know how that happened.
Ed Kravitz.
Well, but Attila Klein walked into my office saying, I think we have a really good graduate student for you. And I said, oh, okay. Do I want this? What do I? And so Judith joined me. Now, when Judith joined me, she was already extraordinarily accomplished. She was halfway through a PhD program. She had a master's, and she learned everything I knew in somewhere between 3 and 4 months. So I quickly learned that the best thing I could do was not get in her way. But we worked together for a lot of the experiments. And Judith would get up in the morning. She would go running. She ran, 6 miles in those days, and would come into the lab and do the dissections. And then we would often sit together at the rig until 10 or 11 o'clock at night, because that was the tradition in those days. You had to put in all your hours. And so we did a lot of those experiments. And I will never forget the day that Judith sat and drew out all the 9 possibilities for the connectivity in the pyloric circuit that was confusing the Jesus. So we looked at him and said, oh my God. And that sort of opened the way to solving the conundrum that led to Judith's thesis papers. The other thing that was really quite remarkable about Judith is she had fabulous hands, but they were also really little hands. And in the days when remember we were using the old Miller killer. So John Miller had designed this device to allow us to do lucere yellow kills of neurons. And this was before there was epifluorescence on the microscopes. So it came in, there was a light source that came in below the preparation. The preparation had to be perfectly situated on a little piece of glass, and everything had to be pinned correctly, and the light had to be focused and all of this. And there was no way I was ever going to get my hands in through this morass to fiddle with this thing. But Judith had these little hands, and she was able to get her hands in to make the Miller killer work. And I swear, only because, show them your hands, only because her hands are really little, did this device that the shop had built with John Miller's designations work. Anyway, it was truly, truly, truly an honor and a privilege to be able to work with Judith early on. And I was never so lucky as to have Judith be my first graduate student, and then Scott Hooper my second. And there's nothing, Scott and Judith were alike in one way or in 2 ways. They were both really fabulous experimentalists, and they were fabulously smart. And in every other way, they're completely different. So it's a great honor for us to, what?
Thank God.
Thank God. Yeah, well, so it's really an honor to have Judith join us today. And she has gone on to do really wonderful work in developmental neurobiology and zebrafish since leaving us. So welcome. It's all yours.
Thank you, Eve. Yes, there are many, many, many stories from those days. But I'm not really going to tell those stories right now, although maybe they'll come up. I'm going to start sharing my screen if I can. Let's see. So can you all see that?
Yes.
Okay, good. So now I can't see you and I can't see the chat. So people should feel free to interrupt me, although I've built in some times for questions too. So when I was a graduate, I guess I will start out with a story. When I was a graduate student, there was something that I bugged Eve about. Well, I bugged Eve about a lot of things, but here's one thing that I bugged her about many times, and that was the question of whether two PDs are the same, and how would we ever know that? Can you guys see my cursor? Yeah, okay. So how would we even ever address that question? So of course, that question has been addressed to a certain extent by people like Dirk Bucher and David Schultz and others, looking at morphology, which is what's shown here, looking at expression of ion channels and other things, both at the protein level and at the RNA level, and seeing that at least morphologically the cells are similar, but not exactly the same. Expression patterns can be different between the two cells. And then I think there's probably still a lot of open questions. I certainly don't know the answer. Maybe some of you do. For example, do two PDs innervate overlapping cells? Do they divide up the territory? If they do that, how do they do that? How are those choices made? So, lots of open questions that probably aren't so easy to get at in a lobster or a crab. So when I went off to work on vertebrates for my postdoc, I was quite nervous because I was very invested in the idea of individually identified neurons, such as the somatogastric, but also many other places in invertebrates that you could study and be really sure you were going back to the same cell in every animal. And so I'm going to tell you some stories from my lab. And the first story is about individually identified neurons in the spinal cord of zebrafish, and what I'm going to call two cats, sort of similar to two PDs. Although in this case, in studying the development of those cells, we discovered that the cells are the same when they start out life, but they actually take on different fates later. And so then I'm going to tell you a couple of other stories. That's the and beyond part of the talk. And one of those stories will be about things that happen in the gut of the zebrafish, innervated by the enteric nervous system, which I suppose you can think of sort of like the somatogastric, although it's got an awful lot more cells in it, and they are certainly not individually identifiable. And then finally, I'll tell you a story about things that we're doing looking at the brain. So it was really exciting when I started working with on zebrafish with this guy, Monty Westerfield. This is an old picture of Monty, although not probably quite old enough. And another guy, Paul Myers, who some of you might know because he's sort of famous for a web presence. He has a website called Pharyngula. And this For those of you who haven't seen a zebrafish embryo, this is what they look like. This is about an 18-hour-old embryo, so you can see the eye. Here's the developing brain, yolk sac that will nourish the embryo until they actually hatch out of the shell that they would be in at this stage and start eating. And these segmentally arranged structures which form the muscle and bone that are used for swimming. So one of the things that we discovered pretty early on is that there are individually identified neurons in the spinal cord. These are motor neurons, and there's a repeating pattern of them. So each of these segments has 3 of these early developing motor neurons. Like any good individually identified neuron, we gave them names, and these cells divide up the muscle territory in a very predictable way. And the other cool thing is that these cells survive through adulthood. They're not replaced by other cells, so they're there in adults, and that's what this looks like. And I'll just point out one other thing. Eve mentioned that I came from Fred Lang's lab, where I was looking at innervation of the tail muscle. And so the innervation pattern of the tail muscle of a lobster is very similar to the innervation pattern of the swimming muscles of a zebrafish. Probably most of you who work on stomatogastric just eat the tails, but the innervation is actually quite interesting. So these cells, these primary motor neurons, the one that I'm going to talk about particularly today is called CAP. Here's what CAP looks like using differential interference contrast microscopy in a living embryo. So you can see the cell body, you can see the nucleus, nucleoli, you can see the axon. This is inside the spinal cord. This is a structure called the notochord, and here's the axon coming down over the notochord to go out and innervate that muscle. And if we put some dye here and let it backfill, we can see that there really is just one cap. This is actually the same cell. But interestingly, we discovered that sometimes there are 2 cells in this cap position. So here you can see them in DIC, and if we put the dye there, here you can see that there are 2 cell bodies. And we worked really hard at trying to find out if there was some sort of pattern here. We looked in all the different segments. We looked on the right side and the left side of animals. We counted them up, and it's actually quite variable. But if you look at a big enough sample, by every measure, whether there's 1 or there's 2, comes up 0.5. So that's random. So we have no idea why sometimes there's 1 and sometimes there's 2. But it turns out, oh, and the other thing I'll say is, well, I'll wait till the next slide. So when there's 1 cell in this position, starts out looking like this. This is what I showed you with the DIC optics, and then this cell grows its axon out and innervates muscle. When there's 2 cells in the position, one of them grows its axon out and innervates muscle. The other one does not do that. Its axon stays here in the middle of the segment. And in fact, most of the time that cell dies. So because the cells look so similar at the beginning, like at this stage, and because another measure of 0.5 is half the time cap would be this cell and half the time cap would be that cell. We wondered if the cells really were equivalent initially. And to do that, we did transplantation experiments. So we took up a donor embryo that was dye labeled, and we could take cap and VAP, and we could, because we can see them, we could do this. We plucked them out and transplanted them. And we did this in 2 different ways. So in half the experiments, we transplanted them into hosts in which the native caps and VAPs had been ablated. And in those cases, both of the cells invariably turned into caps. In the other half of the experiments, we put the cells right next to an existing cap, unlabeled here, but marked with an asterisk. And in those cases, the cells invariably turned into VAP. So that tells us a couple of things. First of all, the cells are equivalent to the beginning. And second of all, there's some sort of interaction going on where it looks like a cap, the presence of a cap, suppresses the ability of a second cap to become a cap. And so we went further and we tested that. And the way we did that was to ablate cap and ask what happened to VAP. And this is the same cell. A few hours later, you can see it's turned into a cap. So that suggests that this cap has some sort of direct inhibitory influence on the ability of this VAP to turn into a cap. But the other thing that I want to point out is that the VAP growth cone always stops here. And not only do zebrafish have individually identified neurons, they have individually identified muscle fibers. So this is a pair of muscle fibers called the muscle pioneers. They're always in this particular location. That's where the VAP growth cone stops. And here's what it looks like in real life. So that suggested to us that maybe there was something important about interaction with these muscle pioneers. So in a series of experiments done with my my previous lab manager, Ali Malangsan, we ablated those muscle pioneers, and then both cells became cap. So we put together a little model. So the idea is if we ablate cap, VAP becomes cap. If we ablate the muscle pioneers, both cells become cap. And that suggested to us that maybe there was some difference, maybe in the timing of when these cells reached the muscle pioneers. One of them signaled to the muscle pioneers, and the muscle pioneers then signaled to the other cell, preventing it from growing a long axon and causing it to become VAP. Okay, so we had a flurry of papers about this back in the 90s, and then we this was always my project. Nobody else in the lab really cared about this. Ellie helped me with experiments, but those of you who are PIs and those of you who work with PIs know that PIs don't get to do experiments very often. So this project kind of went along sort of one paper every decade or so. So the next paper was work done by a graduate student in the lab, Laura Hale, trying to figure out what's important about the muscle pioneers. And so Laura looked at a messenger RNA, shown here in an in situ, for a secreted protein called natrin, which is a protein that's very important in pathfinding, but it's also important in cell survival and cell death. And she saw that it was expressed in these muscle pioneers. And when she knocked down natrin using a morpholino, then that turned into cap, even though the muscle pioneers were still present. So that tells us that the interaction isn't directly between these two cells. The muscle pioneers really serve as an intermediary, but what are they doing? So there's quite a few different natrin receptors. And when Laura looked at the expression of natrin receptors in cap and VAP, she didn't see any difference. Now that doesn't tell us anything about quantification. It was just yes or no. But we really have very little idea of how these cells who start out exactly the same become different from one another and how that plays out. And so, another decade has passed. Well, let me just sum this up. So cap and VAP are initially equivalent. And the big question is, what makes them different? And so another decade has passed, and my colleague, Adam Miller, and I just got a grant a couple months ago to go back to this question, which I haven't looked at for a long time. And our strategy is going to be to do single cell RNA sequencing of caps and VAPs at a variety of stages And we're going to isolate the cells from mutants that we have. So we do have some mutants in which all of the cells become caps. We do have mutants in which all of the cells become FAPs. All of these mutations affect those muscle pioneer cells. So we're really going to try to figure out at the molecular level what is different between those cells, when it becomes different, and how that plays out. And I'll just end this section by saying, I think this is probably something that happens in nervous systems in many different places. Cells that start out initially equivalent diversify, and we really don't have a good idea of how this diversification takes place. And so hopefully this will be a nice place to try to address that question. So I'm going to move on from two caps, and I'm wondering if anybody has any questions before I do that. Okay, not hearing any, I'm going to move on into the beyond. And so part one of the beyond is the gut. So this is a zebrafish at a slightly older age than I showed you previously. This is a larva. And here's the eye, here's the ear, here's the developing gut. And the story I'm going to tell you has to do with the role of the enteric nervous system and mutations that affect the enteric nervous system, and using zebrafish as a model for a particular human disease. This is called Hirschsprung's disease. So, and this work was started by Robert Kelch, who was a postdoctoral fellow in Janni Nusslein-Volhard's lab when they were doing the big screen for zebrafish mutations. So he isolated this mutant in a gene called SOX10, and he brought it to my lab to study as a pigmentation mutant. So the obvious difference between this wild type fish and the SOX10 mutant is that this guy lacks all of its pigmentation except in the eye. You can see these nice pigment cells in a variety of different locations in the wild type. So pigment cells are derived from a set of cells called the neural crest. This is an embryonic population of cells. It migrates all over. It gives rise to skeletal elements in the face. It gives rise to all the pigment cells except the ones in the eye. And it also gives rise to the enteric nervous system. So if we blow up this part of the fish's gut, we can see the enteric nervous system here, a large number of cells, and these SOX10 mutants are entirely lacking in the enteric nervous system. So the enteric nervous system does a lot of different things. It's responsible for a lot of different things in the gut. One of the things it's responsible for is motility, and this is the work of a postdoctoral fellow in the lab quite a long time ago now, Julie Kuhlman. So here you're looking at that same part of the gut. In fact, this picture is from this fish. And you can see this very nice peristaltic-like activity. This has been speeded up a bit. This stuff that's really whizzing around our blood cells, moving in the vasculature. And this peristaltic activity is what's going to propel food that the animal eats from its mouth to its anus. So in the absence of the enteric nervous system, there's a lot of motility, because it's been shown not just in zebrafish, but also in mice and in humans, that there's a lot of myogenic activity in the gut. But this activity is coordinated by the enteric nervous system. So this activity is entirely uncoordinated. And we've shown, although I'm not going to show you, that food does not progress. These guys can eat perfectly well, and the anterior part of the gut up here gets full of food, but it just gets stuck. And that's what happens in human Hirschsprung's disease patients. It's called megacolon. So usually it's not the entire enteric nervous system that's absent, it's only the most posterior part, and the food gets stuck in that posterior part. So the other thing that happens in many Hirschsprung's patients is that they have intractable inflammation of the gut. It's called Hirschsprung's associated enterocolitis. And so Anna Rolig, who was a postdoctoral fellow with my colleague and close collaborator, Karen Gilliman, was the lead on this part of the project. And this is all a collaboration with Karen's lab. And she showed that the zebrafish model also has inflammation. So we look at inflammation by looking at circulating neutrophils. These are innate immune cells, mostly in the vasculature, which you can see here. You can see a few of them in this wild type gut. But in this SOX10 mutant gut, you can see that they're leaving the vasculature and they're infiltrating the gut. And so this is what happens in people, and it's really a problem. Not very much is known about how this occurs. So when Karen came to University of Oregon, she was interested in looking at host microbe interactions. And one of the things that we now know, thanks to her work, is that the vertebrate gut is teeming with microbes. And we're actually able to see them. And this is due to the work of Travis Wiles, who was a postdoctoral fellow in Karen's lab, who figured out ways to actually modify the bacteria in the gut so we can express fluorescent proteins or a variety of other kinds of things. So I'll just tell you a little aside here, being very naive about the microbial world. When I embarked on this collaboration with Karen, I figured, well, it's just bacteria. We know everything about them. And we've got all these sequenced genomes and we can modify them and blah, blah, blah. It's easy to work with. So it turns out that's entirely not the case, except for probably a handful of bacteria, E. coli and a few others that are used in the laboratory. But the bugs in our guts or zebrafish gut are the Wild West. And very little is known about how to culture them, how to modify them. And so it was really kind of a tour de force for Travis to be able to figure out ways to fluorescently label them and allow us to see them. So knowing that there are all these bacteria, we wondered whether they might not be the reason for the inflammation. And so to look at that, Karen's lab developed ways to make zebrafish germ-free. And in fact, with zebrafish, it's pretty easy. Here's the embryo at an early stage of development that has a little eggshell around it. And you can literally just surface sterilize that with bleach and iodine and then raise those fish up in sterile media. So those are the germ-free fish. Of course, we want the control fish to go through the same treatment. So they are sterilized and then they can be inoculated with the microbiome of other fish. And so when Anna did this, first, I'll show you that SOX10 mutants, as I showed you previously, have more neutrophils than the wild types in their guts in the conventional state. But when she made them germ-free, the number of neutrophils went down. So the inflammation is caused by the bacteria. And there's kind of a graded series. So germ-free animals have basically no inflammation. There's no neutrophils in their guts. Wild types have a few, and SOX10 mutants have a lot. So the bacteria are necessary for the inflammation. Anna wondered whether they were sufficient. And to do this, she did transplantation experiments with the bacteria. So she derived donors germ-free, and then she derived recipients germ-free. She derived some donors germ-free, and then she used wild types and SOX10 mutants as donors. So she just took the bacteria out of them put them into these germ-free animals and asked whether that was sufficient to cause inflammation. And what she saw was only the SOX10 mutant bacteria could cause this inflammation. So the bacteria are both necessary and sufficient for this inflammatory process. So that suggested to us maybe there's some big bad bug in the gut of these SOX10 mutants that's really inflammatory and really causing this enterocolitis. And so to look at that, Anna profiled the bacteria from the guts of wild types and SOX10 mutants. And kind of to our surprise, she didn't find any big bad bugs. But what she found was a difference in the abundance of the normal residents. And I'm going to just point out two of them to you. So this green group, the Escherichia shigella group, was significantly decreased, whereas this red group, the Vibrio group, was significantly increased, suggesting that these guys might be anti-inflammatory and these guys might be pro-inflammatory. And so you can test that by just adding the bacteria. You don't even have to add them to germ-free animals. And so that's what Anna did. So here's an animal with its normal number of neutrophils. And what she found is when she added Vibrio, the number of neutrophils went way up. And when she added E. coli, the number of neutrophils went way down. So it's really the abundance of these bacteria that regulates this inflammatory state. And this,
Can I interrupt?
yeah, please do.
What age are you doing this? Is it really, really, really early, or are you waiting a little while?
Right. So the fish are inside of their coria until they're about 3 days old. They hatch out, they start eating at about 4 days. So we do these at about 4 days, and we look at them at about 5, 6, or 7 days. So that's, a zebrafish reaches sexual maturity around a month.
So they're very young.
They're young. They're very young. Yeah, in adults, you lose some of the really nice properties of being able to look at the embryos and larvae. The adults are not optically clear.
Right.
So you can't see what's going on inside of them.
And just as a follow-up question, to go back to the human disease that originally inspired this,
Yeah.
is that, do very young children get sick, or is this an older person disease?
So it's quite variable because the extent of the enteric nervous system that's missing is quite variable. So somebody who's lacking their entire enteric nervous system, that's going to be lethal very soon afterward. But people who are lacking portions of their enteric nervous system, there are some people who are walking around lacking portions of their enteric nervous system. For many patients, what happens is the piece of the enteric nervous system that's not innervated actually has to be resected. So that means that there's motility along the whole length of the gut, because you can just cut off the part that's non-motile, but there's still enterocolitis. So that part of the nervous system that's still there and is sufficient to make the muscle move, maybe not as well as in many people, but sufficient to make the muscle move, isn't able to carry out other important functions. And I should also say that the enterocolitis tends to be episodic, as so many gut-borne diseases are. So people can be fine for a while and then they'll have an episode for reasons that are unknown, and then they'll be fine for a while. And I think one of the reasons that this disease has been so intractable is exactly this. there's not a big bad bug. These are the normal residents of the gut. And for some reason, things become unbalanced. And what reasons can things become unbalanced? I mean, there's so many metabolic and physiological processes, hormonally related processes, stress related processes. You ate the wrong thing that altered your community a little bit. So those are all things that remain to be figured out. But I think this idea that relative abundance of different bacteria can have a huge effect, and you don't need some particularly obnoxious bad actor. And also, I'd say the other piece of this is Vibrio, and we actually know that Vibrio is quite pro-inflammatory, but it's a normal member of that community. It's there all the time. So why is it not causing inflammation all the time? And that's a question we don't know the answer to. So I'm gonna move on to the next piece of it and ask, how does the ENS modulate bacterial abundance? And for this part of it, we've really focused in on Vibrio. This is the work of Christy Hamilton, who is a postdoctoral fellow in the lab until yesterday, and she just got a job and left. So we're very sad and very excited. So Christy actually came to the lab with a lot of experience as a gut physiologist, and she knew that the Vibrio in the zebrafish gut is pretty closely related to the Vibrio cholera that causes cholera, although it doesn't have the toxin. And she also knew that human Vibrios are very good at evading the acidic environment of the stomach. And so they can live in more acidic places. So she asked a really simple question: is the pH of the gut different in these SOX10 mutants? And so she did a really elegant and ingenious, and probably after the fact, very obvious experiment, which is she gavaged fish with pH indicators. So that's the nice thing about something that's transparent and young. You stick a little pipette down its throat and you squirt in pH indicators. And what she found is that zebrafish that are wild type, here you can see the pH indicator in the gut, have a much more basic gut than these SOX10 mutants. They have a very acidic gut. And so here's some quantification of that. And so then she asked what I think is the really important question here. Is this a result of the difference in the bacteria, or does this happen before the bacteria are there? And could this cause the difference in the bacteria? And so she made the animals germ-free, and she saw that the SOX10 guts were still more acidic. So this is a function of the enteric nervous system that we don't know very much about. So it's regulating pH, and then this has obviously big effects in terms of the bacteria. So how's the pH regulated in the gut? So this is the intestinal epithelium. There are proton pumps. They pump protons into the gut, and that makes it more acidic. So if you block these proton pumps, the gut becomes less acidic. And guess what? This is heartburn, and there's heartburn medication out there to make the gut less acidic. So omeprazole, commonly sold as Prilosec. And I will say, during at least the early parts of the pandemic, this stuff was like unobtainable because we all had heartburn. So it turns out you can give this stuff to zebrafish just as well as you can give it to people. And here's a SOX10 mutant with its acidic gut. And here's a SOX10 mutant that's been treated with heartburn medication. So its gut is no longer acidic. And then if we look at the readouts of that, we can see here's the SOX10 mutant with its high abundance of Vibrio, which is very significantly decreased when we give these animals omeprazole. And the number of neutrophils, which is high in SOX10 mutants, normally is significantly decreased also. So it really looks like this acidic environment is sufficient to cause the increase in Vibrio and also the increase in neutrophils. So if we can cure it, can we cause it? And so carbonic anhydrase also regulates the pH of the gut, and carbonic anhydrase can be blocked, which raises the acidity of the gut. And again, there are medications that will affect this. Now, these are not over-the-counter medications, and they're not sold for this purpose because probably nobody wants a more acidic gut. But acetazolamide is sold. It's prescribed as a diuretic and for conditions like glaucoma and epilepsy. And so here's a wild type fish with its nice basic gut. And here's a wild type fish that's been given acetazolamide. And so now its gut has become as acidic as the gut of a SOX10 mutant. Here's the normal distribution of Vibrio in wild types. Here's what happens in these acetazolamide treated fish. So the number of Vibrio goes way up. And that's true also for the number of neutrophils, which goes way up. So really this pH regulation seems to be quite pivotal in controlling the abundance of Vibrio. Now, I certainly don't mean to suggest that pH is the only thing. So we know that the enteric nervous system is controlling motility, it's controlling pH, it's controlling mucus, it's controlling the permeability of these certainly and the ability of things to move between them. And so there are many other aspects that could contribute to this, but pH certainly seems to be an important piece of it. So I'm going to just summarize this by saying, here's a wild type gut with its enteric neurons and its muscle, has a nice microbial community, few neutrophils. Here's a SOX10 mutant gut. It has no enteric nervous system. The muscle moves, but not in a nice rhythmic way. The pH in the lumen of the gut is much higher distribution of Vibrio and many more neutrophils. So in the germ-free condition, we don't see neutrophils in either the wild type or the SOX10 mutants because that requires the pro-inflammatory bacteria, but we do see a much more acidic gut lumen. We can cure this by decreasing the acidity, which decreases the level of Vibrio and decreases the level of neutrophils. And we can cause it by increasing the acidity, and that increases the level of Vibrio and increases the number of neutrophils. So I'm going to end this part of the talk and say very happy to take any questions if there are any, or I'll move on. All right. So the next part of the beyond is the brain. So the last story I'm going to tell you about is how those bacteria, which are here in the gut, affect development of the brain up here. And why would you even think to ask such a question? So the microbiota is linked to a number of developmental conditions that affect social behavior. And social behavior is something that we've been quite interested in. So the nervous system undergoes a lot of postnatal development. So here's a illustration of that. So neurons make exuberant arbors, and then those arbors are remodeled, which is really important for the development of circuitry. Over exactly the same time frame, the microbiota develops and it becomes more and more complex. But there are times when this fails, and that's called dysbiosis. And many people who have conditions such as autism experience dysbiosis. And at least some cases of autism have been linked to a failure of this remodeling process. So we're interested in using zebrafish.
Spartacus.
Pardon? Is there a question? No, guess not. So we're interested in using zebrafish as a model to try to understand how this might work. And so, of course, the first thing we need to have is a readout of social behavior, since that's what we're interested in. This is the work of Sarah Stednetz, who was a graduate student in the lab of my colleague, Phil Washburn, and he's a collaborator on all of the work that I'm going to tell you about. But so here's two zebrafish trying to be social with one another. What you're actually looking at is two fish that are in separate tanks. They're separated by electrochromic film, so we can make that transparent or opaque. And when the fish can see each other, they really want to hang out together and socialize. And the computer can track that, so it can show us where the fish have been in the tank, and it can also show us their orientation towards one another. So they typically orient at 45 or 90 degrees, and that makes sense because their eyes are kind of on the side of their head. So if we want to try to figure out what's going on in the brain, we have to know what part of the brain might be important for this. And to understand this, Sarah took advantage of a lot of enhancer trap lines that were developed by Harry Burgess at the NIH. So what you're looking at, you're now looking at a dorsal view of larval zebrafish, and each of these different colors represents a different enhancer trap that's expressed in a subset of neurons in some part of the brain, and they're hooked up to different fluorophores so that you can see them. So we can also hook these up instead of to fluorophores to nitroreductase, which is a bacterial enzyme. And then if we add the antibiotic metronidazole, it kills only the cells expressing nitroreductase. And so by using a bunch of different lines and killing different groups of cells, Sarah found one particular group of cells in the basal forebrain or ventral telencephalon that are required for this behavior. But of course, that doesn't mean that many other cells aren't required for this behavior, but this group certainly is. So those are the ones we focused in on. So here you can see a control fish, just like the ones I showed you in the previous slide. And here's a fish with ablated V-tail neurons. And you can see they just really don't care very much about being social. Since we're interested in looking at the development of this process, we have to know something about how it develops. And so we know that these V-tail neurons are first visible around the end of the first day of development. And Sarah followed the behavior over time. She saw that it started around here, end of the first week, but it was very robust by the end of the second week. So that's when she assayed the behavior. And because we're interested in looking at the, role of the microbiota for all of these animals, they were derived germ-free, and then they were inoculated with the normal microbiota. And so the rest of the work I'm going to tell you about is a work of a postdoctoral fellow, Joseph Brookman. So here's what two of those 14-day-old fish look like. They were made germ-free on day one, and then they were inoculated immediately with the microbiota. And you can see they behave just like the adults do. So you all probably know that there's this concept in thinking about the development of the nervous system of critical periods. So if something doesn't get wired up at the right time, it's unlikely to get wired up properly later. So because of the timing of the development of the social behavior, we figured that the critical period was going to be sometime in this range. So what we did was we made animals germ-free, kept them germ-free for a week, and then we inoculated them with the microbiota and then assayed their behavior here. And you can see that these guys are really not interested in being social, just like the animal I showed you with the ablated V-tail neurons. And here's quantification of that. Okay, so what's going on with those V-tail neurons that would cause the animals to be unable to engage in the social behavior? And so to look at this, Joseph took a sparse labeling approach. So this is that set of V-tail neurons blown up, and he labeled just a few neurons in each larva so that he could trace their entire arborization. And what he saw, if he looked in the fish that were conventional, he saw that, I'm just showing you one neuron, the neurons had particular arbors, but if he looked in the germ-free animals, he saw a new class of arbors that emerged. So this was not in all of the V-tail neurons, but quite a few of them had these much more expansive arbors. So these are the fish that were made, kept germ-free for a week and then inoculated. And he actually assayed this at 2 time points at 7 days. So while they're still germ-free, and he saw that these arbors were expanded. And then he assayed them after they had been inoculated. And he saw, if anything, the arbors got worse. So as though there really is some sort of critical period for this process to occur. Okay, so how does information get from bacteria here up to the brain to cause a change in the arborization that's going to lead to a change in the circuitry? And so there are a lot of different ways that this could happen. These guys could be secreting various types of molecules that go into the vasculature, and if they can cross the blood-brain barrier, they could act on various cells inside that blood-brain barrier in many different ways. The vagus nerve provides kind of a direct conduit. So the vagus nerve makes connections with the enteric neurons. It also makes connections with a particular set of enteroendocrine cells. These are actually part of the intestinal epithelium, but they're sensory cells. And that information can be conveyed directly to the brain. So that's another possible route. And a third possible route is through the immune system. So I already told you about neutrophils. They're also macrophages, and these are circulating throughout the body. The brain has its own set of macrophages, which are called microglia, that infiltrate the brain early when the brain's forming, and there's essentially no trafficking of those cells between the brain and the rest of the body, at least under normal circumstances. So we focused initially on the microglia. Microglia are cool. They do a lot of things, and one of the things that they do that seemed pertinent in this case was that they're involved in synaptic remodeling. So they prune away excess synapses, and that seems to us to be what had gone wrong in our germ-free animals. And so here's a picture of a microglial cell. These are V-tail neurons. And you can see that this cell has really intimate connections, both with the cell bodies and with the arbors of those V-tail neurons. So perfectly positioned to be involved in this kind of remodeling process. So Joseph counted the number of microglia in the ventral telencephalon, and then he did the same thing in the germ-free animals and saw that there was a decrease in the number of microglia. So, so the number of microglia is being regulated by the microbiota. And so we wondered whether we could make conventional animals look like germ-free animals by decreasing the number of microglia. And to do this, Joseph injected a morpholino, which is an antisense reagent. And this is to a gene called ERF8, which is required for development of microglia. And what he saw is the number of microglia was decreased. And at the same time, when he measured those arbors, they were increased. So it certainly suggests that the number of microglia is important for this pruning process. We're also interested in looking at the molecular level to try to find out what genes and what proteins might be involved. And so to do this, we piggybacked on the work of Michelle Massacoy, who's a graduate student in Karen Gilman's lab, who is involved in a project doing single-cell RNA sequencing and comparing germ-free zebrafish larvae with conventional zebrafish larvae. And so if you're not used to looking at these UMAP plots, this is a two-dimensional plot of three-dimensional space. Each of these clusters has a color and a number assigned by the computer, and the clustering and numbering is based on similarity of gene expression. So each of these clusters represents a different cell type. These clusters are the ones that represent immune cells. And I'll just show you, and this is was surprising, that here's the conventional and the germ-free cells mapped onto one another. And you can see that there are differences in expression, but overall they look kind of the same, which was pretty interesting. So Joseph used 75 core microglial genes and identified microglia out of this region. And then he looked at the ones that are specific for the ventral telencephalon. And so that's this group here. And so one of these genes that really caught our eye is C1q. C1q is part of the complement cascade. And the complement cascade is very important in this remodeling process. So microglia make C1q. They make another protein called C3. They use it to decorate the synapses that they're going to prune, and then that acts through a receptor on those cells. So we found that conventional animals have more C1q and more cells express it than the germ-free animals. The reason you see C1qa and C1qb here is in the lineage leading to zebrafish, there was a genome duplication. So many genes are present in duplicate. And so to ask whether C1q might be important, Joseph knocked it down with a morpholino, didn't see a difference in the number of microglial cells, but he did see an increase in this arborization, providing evidence that the complement cascade is important in this pruning process, and that's likely part, at least, of what's going on in these germ-free animals. So I'm going to end this story here and try to sum up and say what I hope I've shown you today is that the enteric nervous system modulates the intestinal microbial community, and that this intestinal microbial community is important for the number of microglial cells, and it's also important for gene expression of those cells, and that in the absence of the microbiome, or even if the microbiome's been removed and then reinstated, this process doesn't work properly. And so the microbiome is restraining the complexity of these ventral telencephalic neurons by modulating this pruning process. Now, I don't want to suggest that that's the only thing that's going on. There could be direct effects too. We have not looked at those yet, but it wouldn't surprise me, because there's a lot of different kinds of bacteria in the gut, and they can be doing all of those different kinds of things that I talked about earlier at the same time, and all of those can be impinging on the brain in a variety of different ways. So I'm going to stop here and acknowledge all of our funders, the team motor neurons people, the team gut people. I've talked about the specific work of people who were leads on their project. My colleague Karen Gilman, who has been a collaborator on all of this. My colleague Raghu Parthasarathi, who developed the light sheet microscope that we used for a number of those studies. And Team Brain, my colleague Phil Washburn, who's been a collaborator on all of that. Now I'm going to stop talking and happy to take any questions.
Hi, Judith, I have a question.
Yeah.
But first, I will tell you that this is way cool work. Thanks for giving. I either missed it or you secretly didn't address the hypothesis about how the gut communicates with the microglia. How does that signal get there?
Yeah, no, that's a great question. You didn't miss it, and we don't actually know how it gets there. So we don't know what the signal is. So one of the things that we're trying to do is try to work this back to the molecular level. So, there are just huge outstanding questions here. What are these bacteria making? Where does the host experience that? Is it in the gut? Is it in the vasculature? Is it someplace else? How does the host perceive it? So what's the recognition system? And how does the host transduce that into something that affects what's going on in the brain, whether it's the microglia or something else? And those are all things that we don't know, that we would like to know. So we're doing experiments of trying to put back just one type of bacteria at a time and ask what bacteria rescues, because then you can start to ask about the molecular mechanisms that are happening on the bacterial side, which hopefully will lead us to the molecular mechanisms that are happening on the host side. But how the microglia know that there's bugs in the gut or not bugs in the gut, or what kind of bugs are in the gut, is something we don't understand.
Good. So you have more stuff.
We have more stuff to do. Yeah.
Nina.
Fascinating talk, Judith. And Hirschsprung disease is associated also with sudden infant death syndrome and the FOX2B mutation in CCHS.
Yep.
And I always thought it's very simple because you have these FOX2B sensitive neurons that are chemosensors. But now after your talk, I'm thinking maybe it's way more complex, because these vagal afferences go right into the medulla. And I know that if you have inflammation in the lung, you get changes in the brainstem right away within 10 minutes. So the question is also whether really inflammation plays a big role in this, and inflammation has been implicated also.
Hirschsprung's disease is interesting because there's many loci that can cause it. So SOX10 is a big one, FOX2B is another one, RET, but there's a whole lot of other loci that can cause it, and it looks different in different with different mutations or different alleles of different mutations, or if people are carrying two different alleles, two people who are SIBs and carry the same alleles may have very, very different presentations. So there's an enormous amount of complexity. And I think part of that enormous complexity could have to do with not just the mutations, but all the physiology that then happens around those mutations, all of the things that are happening in the gut, changes in the microbiota, changes in the immune system. I think immune cells, and there are, kind of private immune cells for different parts of the body too. And I don't know, but I would guess that just like in the nervous system, some of those immune cells may have critical periods where if they don't undergo some particular process at some particular time, it's not going to happen and they are not going to mature properly. So yeah, it'd be nice to think that it could be simple, but I think it's really not, and hard to study. And we're picking it apart one little piece at a time.
By the way, I just heard a talk where like if the microglia get activated, they get attracted by neurons that are bursting.
Yeah.
And so it's really like a whole dynamic interaction. Yeah.
Yep.
So, Ron, did you have a question?
Yeah, I did. I was just wondering, I don't know if I missed this. Are there microglia in the gut? And are there less microglia more microglia in the gut when there are less in the brain?
So no, so microglia are derived from the same lineage as macrophages. So the macrophages are circulating immune cells. We all have bazillions of them and they turn over at some rate. So microglia, so in vertebrate animals, the brain starts as a flat sheet of some sort and it rolls up and then it develops this blood-brain barrier. Happens in different ways in different vertebrates, but the microglia are derived from macrophages that enter it before it's completely rolled up. So this is a private population of macrophages that generally does not traffic into the rest of the body. They're segregated into the brain. Now, I think we're learning more about these cells and learning that there is some trafficking and it is very gated and it happens under particular circumstances. But I think in bulk, there's not trafficking. So the ones that are in the gut and are part of what's circulating in the body they're completely divorced from the ones that are in the brain and have been so since the beginning of those or early in those lineages. The other thing that we're learning though is that there are private groups of macrophages in the gut. So there are some in the muscle cells, they're called muscularis macrophages, and they have some particular jobs to do that are probably different than the ones that are in the regular circulation. And I don't think we know very much about that at this point. It's probably the case that there are private collections of macrophages in many other parts of the body that kind of hang out in, the liver or the pancreas or wherever it's going to be and don't circulate very much. I don't think we have yet the tools to recognize them. And this is far better studied in mice than it is in zebrafish. But of course, mice are not transparent and people don't do very many studies in mouse pups. They usually wait till they grow up to be adults and then you infer the developmental process without having watched it. You just see the endpoint. So trying to understand how all of these populations develop and how they're related to one another, I think is kind of a new big thing.
So Judith, have you been able to or tried or thought to get some wild caught animals from places in India, for example, and to see how different the bacteria are, the microbiome is, or what influence that would have on brain development?
So that was done. I have to say that's almost an impossible experiment to actually do, just because even in the pre-pandemic days, importing and exporting animals,
I understand.
this unbelievable red tape. Anyway, animals were caught in India, and their microbiomes were profiled and compared to animals from different facilities in the United States. And what you could see is there's kind of a core microbiome, but it's not, it's not identical. And you could also see the history of, so most of the fish in facilities in the United States, one way or another, either came from the Oregon zebrafish facility, or they came from Yanni Nussland Lohard's facility in Tübingen, Germany, and those were fish that originally came from hobbyists, fish hobbyists. And so you could kind of see the lineage of those fish by comparing the microbiomes and changes in the microbiomes. Now, what those changes mean, we have no clue about that. And of course, nobody's ever looked at the brains and tried to correlate any of these things or looked at social interactions. But I mean, you can totally imagine the different populations of fish that out in the wild that school differently. Could that involve differences in their microbiome? Sure, but it's going to be a hard question to actually get at.
So many of you remember Vatsa Thermulai, who is back in Bangalore working on zebrafish. And when she first went back to India, she had so much trouble importing lines that she actually had to catch wild type in India. And then of course, so she had her own colonies that she was breeding. And I wonder if you've ever talked to her about, are there sort of lab to lab differences that people have not known what to attribute to?
There are not at that level, because so much of what people are doing is in embryos, and the embryos don't really have a microbiome yet. They become colonized when they start to eat. Of course, mom's microbiome could have an effect on what happens, in the formation of that egg. But people haven't observed that level of difference. But I will tell you one interesting story is that in zebrafish domesticated by George Streisinger in Eugene, they have lost the major sex determining locus. They still make males and females, but that major locus, which is found in wild fish, has been lost. In fish domesticated in Yanni's lab by Mary Mullins, they have also, and these are fish from different sources and many years apart, they've also lost the same locus. So domestication of animals can make an enormous difference. And certainly people who study mice see this. Some postdoc will get all their mice from Jack's, and they'll then go off to their job and have a new facility, and the people at the new facility get all their mice from Charles River, and they'll get all their mice from Charles River, and they'll go, oh yeah, my whole thing that I studied, that I wrote my thesis on, isn't happening anymore. And people have made some really major discoveries, mostly about the immune system, but also about microbiota, based on those kinds of turned out to be wonderful accidents, but in the beginning they seemed to be horrible accidents. So, you know, there's just broad swaths of biology we just don't know anything about. And I would guess that many things that people study are affected by the microbiota, and that we will learn many more subtleties about some of the things that we study that we never thought the microbiota were involved in, once we know enough to think about how to pay attention to that. Right now, I don't even think we have the tools to know how to pay attention to that.
More questions, comments? No?
Thanks, Judith.
Thanks,
It was-
Monique.